Everything you know about melatonin is about to change. Okay, maybe not everything, but what if I told you that melatonin is not just a sleep-inducing molecule? Surprised? Well, I sure was when I first found this out. You see, the past evidence has led us to believe that melatonin's primary and oftentimes sole role in the human body is to make us fall asleep. And while this is a role melatonin does play, it's only part of the full story behind this fascinating molecule.
And indeed, all of the melatonin in your body, at least to my knowledge, comes from the pineal. There might be small amounts secreted from other areas of the brain and body, but that's largely the case, that it comes primarily from the pineal.
Get this, melatonin that makes us fall asleep only accounts for 5% of the melatonin in our bodies. 5%. Crazy. Okay. Here we are going to get into all the details, but I want to leave you with this one little sneak peek. Melatonin, the type that makes us fall asleep, is produced in the absence of light. Many of us may know this, so no surprise there. But what if I told you that the majority of melatonin is actually produced in response to light from the sun? Crazy, I know. And I wish I could tell you the whole story in 60 seconds, but there's a lot of fascinating detail I want to cover. So stay with me here as we uncover the intricacies of melatonin. Today, I want to dive deep into a paper published in 2023 by 4 premier melatonin researchers. The paper is titled this: Melatonin, both a messenger of darkness and the participant in the cellular actions of non-visible solar radiation of near-infrared light. Let's dive in. Before we get into the really good stuff, not to say this stuff isn't good, I want to first bring you on a short biology lesson. So, you have all heard of the term protein. Proteins are essential to all forms of life, hence why they're considered the basic building blocks of life. Chains of amino acids strung together to form a structural unit called a protein. These amino acids can be split up into 2 distinct groups: essential amino acids and non-essential amino acids. Essential amino acids means our bodies cannot produce them on its own, while non-essential amino acids our bodies can produce. For the sake of this video, we will be highlighting one specific essential amino acid. That amino acid is tryptophan. You see, the reasons humans can't make tryptophan is because the chemical pathway to do so is not present in human cells. This pathway is named the shikimate pathway. Okay, now why are we talking about a simple amino acid when this video is supposed to be about melatonin? Well, melatonin is derived from tryptophan, meaning in order for melatonin to be produced, your body first needs to start off with tryptophan. Before we go any further, I want to take you down a molecular cascade of events, showing you exactly how tryptophan gets converted to melatonin. Now, this may seem like a meaningless extra information, but rest assured, there is a reason I want to run you through this. It will be key for you to understand the full picture of melatonin. Okay, fasten your seatbelts because we are about to get a little technical. First, tryptophan gets hydroxylated by the enzyme tryptophan hydroxylase. You see where it gets its name from. Okay, moving on. This new hydroxylated tryptophan, or what's now called 5-hydroxytryptophan, undergoes another reaction called decarboxylation. Here, the enzyme AADC removes a carboxyl group, converting 5-hydroxytryptophan into 5-hydroxytryptamine, or what most people call serotonin. Serotonin is then converted into NAS, or NAS. This conversion is only made possible by the enzyme acrylocalamine N-acetyltransferase, or AANAT. This is key. AANAT steals the acetyl group from acetyl-CoA, adds it to serotonin. The end result of the reaction is the molecule NAS, or NAS. Okay, stay with me here, because we are only one reaction left until we finally meet our good old friend, melatonin. Here we go. NAS, or NAS, gets methylated from the kindness of our friend Sam. Sam donates a methyl group to NAS with the mutual friend or enzyme N-acetylserotonin O-methyltransferase or ASMT. We did it! We now have our friend melatonin. Before we dive deep into new research, let's establish what scientists have known for a while. As mentioned earlier, 5% of melatonin that is produced in the body is used to help us fall asleep. This melatonin is called pineal melatonin, for it is produced in the pineal gland in our brain. And before we move on, I just want to preface you that you will hear me say both pineal and pineal, two different pronunciations for the exact same thing. So apologies in advance. Let's take a look at this diagram to get a full picture. During the day, we are exposed to light. That light ranges from the ultraviolet to the visible, which encompasses all the colors of the rainbow, all the way to the infrared, an invisible light wavelength. During the day, short wavelength light, specifically 480 nanometer blue light, inhibits melatonin production. This blue light enters the eyes and gets absorbed by a pigment called melanopsin. Melanopsin is found in these special cells in our retina called intrinsically photosensitive retinal ganglion cells, or IPGRCs for short. IPGRCs are non-visual photoreceptor cells, meaning they do not assist in the visual function. Instead, they communicate directly to our body's internal timekeeper, our internal clock. As you can see in this diagram I drew here, that light sends a signal down the retinohypothalamic tract, or RHT, to the suprachiasmatic nucleus, or CSN. You could think of the CSN as your body's Big Ben. Yes, I know. If you're hearing this for the first time, it may sound a little crazy, but your body has a clock tower in its brain. Just like the Big Ben, the clock tower tells everyone, all your cells, what time it is. Once the chemical signal reaches the SCN, the suprachiasmatic nucleus, the SCN relays the signal to the paraventricular nucleus, which transfers the signal to the intermediolateral cell column, or IMCC, and further down to the SCG. Here, the signal of light tells the brain to shut down these postganglionic sympathetic fibers you see here, and norepinephrine is then blocked from being released into the pineal gland, which results in no melatonin to be produced. At night, in the absence of light, thus the absence of the signal pathway, the postganglionic sympathetic fibers do not get suppressed, and norepinephrine is released into the pineal gland, starting the production of melatonin. This pineal melatonin, produced in the cytosol of pinealocytes, discharges out of the pineal gland and into the cerebrospinal fluid, the fluid that surrounds your brain, bathing your brain in melatonin and helping us fall asleep. Wasn't that fun to go through? I think it was. Or maybe just because I got to draw such a beautiful and very realistic diagram of the process. Either way, before we go on, I want to do a 2-sentence recap. So, melatonin, synthesized by the amino acid tryptophan, is released from the pineal gland into your brain and bloodstream at night in response to darkness. For this reason, melatonin has been long considered the chemical expression of darkness. We should note, again, that pineal melatonin, everything we just covered, only accounts for just 5% of the total melatonin made in the human body. The other 95%, for the sake of simplicity, we will call this extra-pineal melatonin. Extra-pineal meaning beyond pineal. For the remainder of this video, we will be covering everything you need to know about extra-pineal melatonin. In this next section, I want to surprise you with something shocking. Melatonin is the strongest antioxidant any organism has. A quick nuanced point here. Both pineal and extra-pineal melatonin can act as an antioxidant, but pineal melatonin's antioxidant properties are secondary to its sleep-wake cycle properties, whereas extra-pineal melatonin's antioxidant properties is its primary property. There are 3 unique characteristics of melatonin that differentiates itself from all other classical antioxidants. I will quickly cover the most important one. Here it is. One molecule of melatonin may have the capacity to scavenge up to 10 reactive oxygen species. For those who don't know, reactive oxygen species, or ROS, are these molecules that are generally thought of as damaging and cause oxidative stress. Although this is nuanced, we can think of them generally as bad. So, while melatonin can scavenge up to 10 reactive oxygen species, classical antioxidants like vitamin C, vitamin E, coenzyme Q10, and glutathione typically only detoxify one free radical per molecule. What's interesting is that the mechanism for this tenfold increase in antioxidant capacity from melatonin stems from the metabolites generated by melatonin. Melatonin reacts with many different reactive oxygen species, and the result of these reactions are new and different molecules. These new and different molecules, or metabolites, have the capacity to function as free radical scavengers themselves. So, melatonin can scavenge 10 times more reactive oxygen species than classical antioxidants. For this reason, and a few others, melatonin is considered one of the world's strongest antioxidants. By now, you've learned that pineal melatonin is produced in the pineal gland. But where is the 95% of the rest of the melatonin produced? Well, don't I just have the answer for you? Originally, it was believed that all melatonin was synthesized in the cytosol of the cell, the area inside the cell between the little organelles. However, there was very little data supporting this claim, and it is now believed through strong amounts of evidence that melatonin is primarily produced within the mitochondria of practically every cell in the human body. This assertion is supported by a multitude of evidence, including, but not limited to, the following. First, the rate-limiting enzyme of melatonin synthesis, as previously discussed, is AANAT. AANAT is found to be exclusively housed in the mitochondria. So, if AANAT is needed for melatonin synthesis, then the melatonin must be synthesized where AANAT is localized. Second, in addition to AANAT, the other enzyme needed for melatonin synthesis is ASMT. Both AANAT and ASMT, as well as their mRNAs, are localized in the mitochondria of plants and many animal cells. Third, melatonin has been directly measured in the mitochondria of liver cells. Fourth, extremely high levels of daytime melatonin were observed in the mitochondria of rat heart cells, rat liver cells, and rat brain cells. 5. In addition to melatonin, its metabolite or breakdown product, AFMK, was also identified in the mitochondria. The sixth piece of evidence comes directly from evolutionary theory. The evolutionary precursor to mitochondria, alpha proteobacteria, have the capacity to synthesize melatonin. So, this process of mitochondria producing melatonin seems to be an evolutionary conserved mechanism after the endosymbiotic event. 7. Isolated mitochondria from mouse oocytes, which are the ovarian cells, and apple tree leaves both synthesize melatonin. There was one really fascinating thing about the oocyte study. When the mitochondria were incubated with serotonin, a precursor to melatonin, melatonin was synthesized. However, when serotonin was not present, melatonin was not formed. Now, these mitochondria from the ovarian cells will eventually be the mitochondria of the offspring, making it very, very likely that every one of the offspring cells will retain the ability to produce melatonin. 8. Researchers have identified melatonin in the mitochondria of human salivary gland cells. And finally, the best for last, the ninth piece of evidence, and again, this is not an exhaustive list, brings us back to two enzymes, ANAT and ASMT. These two enzymes, let's just call them melatonin-required enzymes, have been detected in the brain cell mitochondria, where virtually absent in the cytosol. Interestingly enough, in cells of the pineal gland, the expression of one of the melatonin-required enzymes exhibited a circadian rhythm. However, there was no circadian rhythmic pattern exhibited in the non-pineal gland brain cells. This provided strong evidence that the continual production of melatonin in the mitochondria of neurons most likely provides constant protection to these cells from oxidative stress. This melatonin serves a distinctly different function from that produced in the pineal gland, which primarily conveys photoperiodic information. With all this now understood, the available evidence strongly suggests that melatonin is synthesized in the mitochondria. And taking this a step further, in the mitochondria are virtually all cell types which contain mitochondria. But why is this important? You see, our mitochondria undergo cellular respiration, which is the process by which they produce adenosine triphosphate, or ATP. ATP is considered the energy currency of the cell. During this process, the accumulation of reactive oxygen species, or ROS, and reactive nitrogen species, RNS, are produced. Elevated ROS and RNS are drivers for oxidative stress within the cell. Thus, the locally produced melatonin in the mitochondria acts as on-site protection against these free radical molecules. Additionally, acetylcoenzyme A, acetyl-CoA, which should ring a bell from the rundown of the melatonin synthesis pathway and reactions, is itself synthesized, guess where? In the mitochondria. And as we mentioned earlier, acetyl-CoA is an essential substrate for AANAT, for melatonin synthesis, making melatonin production in the mitochondria very, very efficient. Okay, I want to add this for a quick aside, and then we will discuss mechanisms by which melatonin production is stimulated. Here it is. When a cell turns cancerous, the cell switches from cellular respiration to cytosolic glycolysis, also known as the Warburg effect. This, in turn, results in less acetylcoenzyme A production in the mitochondria. With less acetyl-CoA, less melatonin is produced, which is evident by the fact that mitochondria of cancer cells have about half the melatonin compared to the normal cells. Also evident by the fact that cancer patients have lower levels of melatonin. Okay, that aside is super, super interesting, but a story for another time. Now, on to what we have all been waiting for. This is the key point I need you to understand, is this. As mentioned earlier, the reference to melatonin being the chemical expression of darkness has been widely accepted since pineal melatonin has been the focus in melatonin research, and it is only produced at night. However, through new advancements in melatonin research, this statement must be modified. In addition to pinealocytes, as mentioned, it may be the fact that all other cells synthesize extra-pineal melatonin, which is not suppressed by light, has no circadian rhythm, and is actually enhanced by light. You heard that right. Extra-pineal melatonin, made in our mitochondria, is produced in response to light. Before we go on, I do want to add the caveat that none of the following information is mere conjecture. In fact, all of it is based on recent accumulated evidence. However, much of it is extremely understudied and may need further refinement as new evidence arises, just like anything in science. That said, I will do my best to provide you with all the evidence we currently have. So here we go. So, I mentioned to you that this extra-pineal melatonin is synthesized by the addition of light. So, which type of light, you may ask? It's infrared light, specifically near-infrared light. And let's stop right there really quickly and bring you up to speed on light. Maybe not as fast as the speed of light, but it will be pretty fast. Here we go. While it may seem like a simple thing to understand, I want to make sure you get the full picture. Let's start here. Humans evolved under what's called full spectrum sunlight. The full spectrum encapsulates the following: ultraviolet A, ultraviolet B, violet, blue, green, yellow, orange, red, near infrared, and infrared. UV, ultraviolet, and infrared, IR, are both invisible light wavelengths to the human eye, meaning the human eye cannot perceive UV and IR. To get a better grasp, about 50% of sunlight is infrared, 40% is visible, and less than 10% is UV. So the majority of light coming from the sun is in the infrared portion of the spectrum. The point I want to make here, and we will cover this at the end, is that our current light environments, where people are spending 93% of their daily lives indoors, are almost completely void of infrared light. Now, that should give you enough of an understanding for the following. Drawing from this 2019 paper titled Melatonin and the Optics of the Human Body by Scott Zimmerman and Russell J. Ryder, we now know that near-infrared light can penetrate the human body at least 3 inches deep, and through optical models, interact with virtually all of our cells. Our bodies also have the mechanism by which it can propagate this infrared light through specific areas, such as the cerebrospinal fluid in our brain, the amniotic fluid, and blood vessel walls. In contrast to blue light, which only accounts for 2% of the solar spectrum, is absorbed by our natural environment and barely penetrates the skin, near-infrared light flips all those things completely upside down. Near-infrared light represents the majority of the solar spectrum. It is reflected by our natural forest environments, and it penetrates deeply into the body beyond the skin. In fact, the beneficial effects of red and near-infrared light on human health has been extensively studied. And while this is a video for another time, the point to make is that these longer wavelengths of light, red and infrared, have profound and seemingly infinite effects on our bodies. It has been proposed that some of the beneficial outcomes seen in these so-called photobiomodulation studies using red and infrared light are, at least in part, due to the synthesis of melatonin within the mitochondria from the red and infrared light exposure. It has been well established that many of the beneficial outcomes from infrared light treatment are similar to those of melatonin treatment. For this reason, the authors of the paper propose a mechanism by which near-infrared light treatment impacts melatonin synthesis at the cellular level. Let's take a look at this proposed mechanism, keeping in mind that it is a speculative pathway and not fully confirmed by studies. As near-infrared light from the sun penetrates the skin and propagates deep into the tissues of the body, it penetrates the cell and reaches the mitochondria. Within the electron transport chain of the mitochondria lies cytochrome C oxidase, the primary photoreceptor. Cytochrome C oxidase absorbs the near-infrared photons and undergoes a conformational change. This structural change releases nitric oxide into the mitochondrial matrix. That nitric oxide then activates soluble adenylyl cyclase, converting ATP into cyclic AMP or CAMP. Elevated CAMP activates protein kinase A or pKa, which then phosphorylates AANAT. The phosphorylated AANAT now has the ability to help synthesize melatonin via the same exact melatonin synthesizing pathway we learned about earlier in the video. Now, again, this pathway still needs to be validated, but if correct, it provides the mechanistic pathway by which near-infrared light stimulates melatonin production within the mitochondria of every single cell. Now, this proposed pathway wouldn't exist without the observational evidence of near-infrared light having these capabilities of increasing melatonin. So let's look at some of this evidence. We're going to start off with the weakest evidence to the strongest evidence. Here we go. 1. Melatonin has been shown to increase in response to near-infrared light. 2. Exposure to red infrared heat lamps increased salivary melatonin levels in piglets when compared to exposure of heat lamps without near-infrared light. 3. In female athletes receiving full-body red light treatment for 30 minutes per night for 14 days straight, their morning serum melatonin levels were doubled compared to the control group. It should be noted here that the participants who got the red light exposure had their eyes covered with very dark glasses, allowing for the speculation that the increase in melatonin was not of pineal melatonin, but of extra-pineal melatonin through the skin exposure since the eyes were covered. 4. As you can see in this graph, data from participants exercising outdoors shows large quantities of melatonin surging within the first 20 minutes of strenuous exercise outdoors. Shown in the graph, there was a greater than 5 picograms per milliliter minute ramp up rate during the sunlight exposure group, compared to only 0.15 picograms per milliliter minute of melatonin ramp up in the participants who were under dim indoor lighting without infrared light. Some of the data showed that sweat melatonin was independent of plasma melatonin levels, suggesting that independent sources, thus not being from the pineal gland. However, The many confounding variables limited the conclusions that we can draw from this graph. For instance, is the increase in melatonin solely due to near-infrared light? Is it from UV light? Is it from green light, from red light? We also do not know if this melatonin spike is due merely to exercise and the increase in reactive oxygen species that we get from exercise. Would we see similar results by simply laying outside without exercise? We simply do not know. And as always in science, more research here needs to be done. So, those are the main pieces of evidence we have currently demonstrating near-infrared light has the ability to increase melatonin production. And while I really wish I could provide even more evidence, the studies are simply not there. And there we go. You now know more about melatonin than 99% of the world's population. Okay, no, I don't have a study to back that fact up, but it's probably accurate to say. Now, in closing, I want to provide a few practical takeaways from this video which are supported by current scientific data. The first and foremost is to get outside more. Whether it is in direct sunlight or under shade, our bodies are meant to be outside. In doing so, you will be surrounded by infrared light, which has a boatload of benefits, some of which have been covered today. If you can't get outside, I highly recommend purchasing an incandescent light bulb to place on your desk or somewhere in your indoor environment. Quickly, I will show you mine. Here's an incandescent light bulb that I use, and bringing this back into your indoor environment will help add back in these longer wavelengths of red and infrared light. This is crucially important to have. And lastly, at night, once the sun sets, be sure to dim the lights in your room. If possible, purchase a red, ideally incandescent, light bulb to turn on instead of the classical overhead LEDs. You can see mine right over there. Another great option, and I also suggest everyone to get one, is a pair of blue light blocking glasses. Blue light blocking glasses eliminate most of the stimulating light wavelengths, specifically the shorter wavelengths of blue and green light, preventing the signal to your intrinsically photosensitive retinal ganglion cells and allowing your body to produce pineal melatonin. Okay, that is it. 3 practical takeaways you can start implementing today. If you're still here, I want to thank you for listening to this video all the way through. I truly hope you got some value from it, and I think you did. This being my first ever long format video, I'm really looking for any critique and any form of suggestion to be implemented for my future videos. So if you have any suggestions, please feel free to comment directly on this video or message me on my Instagram linked below. As always, keep questioning, stay curious, and take steps towards a healthier